Human impact reaches farther than once believed. The Deep-Sea Debris Database by Jamstack reviewed 5,010 deep-sea dives and identified approximately 3,425 human-derived debris items, mostly plastics, bags, wrappers, fishing lines, many at depths exceeding 10,800 meters. The deepest documented plastic item was found at 10,898 meters. Microplastic concentrations in water column samples near the bottom have been measured at 2 to 13 particles per liter, varying by location and sampling method. Why do these numbers matter? Because the most valuable thing discovered at that depth isn't treasure or metal. It is life in extreme forms, especially these microorganisms. They represent a genetic reservoir of enormous potential. In terms of bioprospecting, the frequency of biosynthetic gene clusters in Hadal microbial genomes is significantly higher per genome than in shallow marine bacteria. For instance, the average number of predicted novel BGCs per metagenomic bin in the 10,600 to 11,000 meters range is approximately 12 to 15, compared to 3 to 5 in coastal bacteria. Those gene clusters could encode new antibiotics, anti-cancer compounds, stress tolerance proteins, or industrially useful enzymes. With antibiotic resistance causing an estimated 700,000 deaths per year globally, the discovery of even a single broad-spectrum antibiotic candidate from Hadal bacteria could save millions of lives. Industrial enzymes that can be used at low temperatures and high pressures save energy and significantly reduce industrial energy costs when chemical processes can be carried out at 2 to 4 degrees Celsius instead of 30 to 40 degrees Celsius. Bioremediation involving plastic degradation enzymes from these microbes, assuming scale-up, could reduce plastic accumulation by millions of tons per year. The Mariana Trench is not just the deepest place on Earth, it is perhaps the last frontier of life as we know it. Lying in the western Pacific Ocean, east of the Mariana Islands, this submarine canyon is approximately 2,550 kilometers long, 69 kilometers wide at its broadest, and plunges as deep as 10,984 meters. That spot, called Challenger Deep, is the greatest known depth in all Earth's oceans documented by modern sonar and manned robotic vehicles. To put this into perspective, even if Mount Everest, which stands at 8,848 meters, were turned upside down and placed inside the pit, it would still be approximately 2,000 meters underwater. The ambient pressure there is staggering. Approximately 1,086 to 1,100 atmospheres, which is over 1,100 times the pressure at sea level. Equipment must endure literally tons of force per square centimeter. Our understanding of Challenger Deep expanded in 1960 when Jacques Picard and Don Walsh in the Trieste dived to 10,916 meters. That dive took about 4 to 5 hours, including descent, bottom time, and ascent. It was the first time humans physically touched the deepest mapped point in the ocean. Almost 52 years later, James Cameron piloted Deep-Sea Challenger solo, reaching 10,908 meters. Then, in 2019, Victor Vescovo's vehicle Limiting Factor, part of the 5 Deeps expedition, recorded 10,928 meters in Challenger Deep, one of the most precise modern measurements. These dives are rare. As of 2024, manned or robotic visits to depths below 10,500 meters number fewer than 20 successful missions. Only a few research institutes, Woods Hole, 5 Deeps, and China's deep-sea programs have the funding and technical capacity. Costs of single deep dives can reach tens of millions of US dollars once you include design, engineering, logistics, live support, recovery of samples, and processing. Despite the extreme pressure and near-freezing temperatures, life does exist. In sediment cores and water samples from 10,000 to 11,000 meter depths, scientists have detected microorganisms, bacteria, and archaea capable of surviving under anaerobic conditions with extremely low nutrient input. In the 2025 MIR dataset, researchers collected between 1,600 and 1,700 sediment samples at various depths between 6,000 and 11,000 meters. From those, they generated approximately 92 terabases of metagenomic data and identified 7,564 distinct microbial taxa about 89% of which were previously unrecorded in public databases. Let me give you an example. In a sample from 10,900 meters, researchers found gene clusters associated with polyketide syntheses, complex enzymes that often play a role in antibiotic production. Also discovered were novel proteases that remain functional under high hydrostatic pressure and low temperature. Enzymes that under lab conditions at 2 degrees Celsius still retain over 60 to 70% of their activity compared to their optimal states. Another noteworthy macrofauna record is that the Mariana snailfish was observed at a depth of 8,178 meters in 2017. Some recent reports indicate similar or slightly deeper observations in neighboring trenches. These fish have physiological adaptations such as reduced bone mineralization, gelatinous body structure, and membrane lipids that maintain fluidity under pressure. In genetics and evolutionary biology, the depth-stratified datasets show that microbial diversity does not simply decline with depth. Below 10,000 meters, new clades appear that are phylogenetically distinct. These extremophiles challenge previous models that assumed severe decline in diversity with depth. The mere dataset's richness and evenness metrics remain surprisingly high even at 11,000 meters. Yet, the value comes with fragility. Many hadal organisms are slow to reproduce, have long life cycles, and low population densities. Sample collection is destructive. Plastic pollution infiltrates even bottom sediments. The presence of chemicals, heavy metals, persistent organic pollutants, has been detected in seabed sediment cores at depths over 10,500 meters. These pollutants can bioaccumulate. International scientific policy is responding. By 2026, proposed treaties aim to establish deep ocean sanctuaries in regions including the Mariana Trench. Research permits may be limited. Environmental impact assessments mandatory. Open data mandates are being adopted by MEER, 5 Deeps, and other projects. Technology investments are accelerating. New unmanned landers capable of taking sterile sediment cores at 11,000 plus meters. In situ, DNA extraction and stabilization tools. Real-time pressure sensors measuring chemical fluxes. Budget commitments for such deep-sea infrastructure are in the hundreds of millions of dollars globally. When you weigh the evidence, what emerges is clear. The most precious discovery in Challenger Deep is not precious metal, but life's persistence in extreme adversity, particularly the microbial genetic diversity, biochemical ingenuity, and evolutionary novelty. These life forms are priceless in terms of what they offer: medical breakthroughs, environmental remediation, understanding the limits of life, and even guiding the search for life beyond Earth. The irony is sharp. The very same environment that holds this treasure also shows how far human pollution has spread. Plastic waste at nearly 11,000 meters, microplastics per liter of water near the seabed, persistent toxins in sediment cores. It's a warning. If we don't protect these extreme ecosystems, not only do we lose potential solutions, we may irreversibly damage ecosystems we barely understand. So the real question isn't just what is the most valuable thing in the deepest place on Earth. It's whether we are ready to recognize its value, protect it, and responsibly harness its potential.